This week we're talking about cell theory and the scientists that contributed to cell theory. And so the thought we started out with today was this, that cell theory states, in part, that all living things are made of one or more cells. This very important part of biology took 200 or so years of observations, experimenting, scientists working together. So what we're going to do now is we're going to list and explain some of the contributions of the cell dudes, that's the cell scientists, and talk about why it did take 200 years for something that seems so simple and even obvious to us. First of all, let's back up a second to yesterday's worksheet about um, yesterday's worksheet where we talked about what is cell theory. Because before we can actually talk about cell theory, we should probably look and see what it is. Yesterday, we talked about cell theory. The three main parts of cell theory are that all living things are made of cells, cells are the basic unit of structure and function in all living things, and that all cells come from other cells. So, as I was talking about this during second period, a couple people were asking, well, if it's a theory and you have proof, well, basically they're confused about the difference between a hypothesis and a theory. And so that's a good point to bring up right now because in science, the word theory is used very differently than in kind of everyday life. So in science, what's a hypothesis? You guys know what's a hypothesis? Hypothesis is what? Yeah, so a hypothesis would be an educated guess or a prediction based on evidence. A hypothesis is testable and can be proven or disproven. The problem is that in everyday life, when people say, oh, I have a theory about that, really what they mean is they have a hypothesis. And some days when I get annoyed about this, I correct people, but they don't really find it very amusing. But they say, oh, I have a theory about what that happens. You watch TV, movies, everyday life, that's what they say. What they mean is they have a hypothesis. Because in science, what were you going to add? Okay. So in science, in fact, a theory is well accepted. It's proven by experiments. It has evidence to back it up. In science, the hypothesis is the educated guess that you can prove or disprove. And then once it has lots of evidence, it becomes a theory. So in that way, cell theory, we all agree living things are made of cells. That's very logical. People all over the world, like I said, biology is based upon the fact that things are made of cells. Now, could a theory ever change? You know, it could if there's enough evidence or questioning. Probably this theory would never be thrown away, but in some of the classes we've talked about viruses are not actually made of cells. Should viruses be considered alive? You could really argue either way. And part of the reason viruses are not included in this original definition, in the original 6 characteristics, is because they are so small, they were impossible to see at the time 300 years ago that this was made into a theory. So a theory might change over time, slightly. Usually it's not totally thrown away. The only thing more strong in science than a theory is a law. So we have the laws of physics. There's a few other laws. In biology, we don't usually have laws just simply because the life is kind of dynamic and changing and hard to measure. Okay, so there we go. Now let's jump ahead, jump back to where we were. These are the scientists we're going to look at today. For the quiz on Friday, you do need to know a little bit about each one of these scientists. So that's what we're going to do now. And I don't care if you know exactly their nationality and what year and all that sort of stuff, but you should know what major contribution did they make. So let's start with Robert Hooke and what he found. So let's go to our first group, and you can tell us about Robert Hooke. All right, so Robert Hooke, he saw the first cells in cork bark, and he named them cells. He came up with the name cells, which means little empty rooms, like a prison cell. And the kind of ironic thing is that the cells he saw, he is famous basically for discovering cells, but the cells he saw were not alive. They were empty, dead bark cells. And this was around 1660. All of these discoveries, the reason why we talk about all the different places these people are from is just to emphasize how many different people contributed to this scientific theory, and that by the time we get cell theory, it's been almost 200 years from him seeing the first cells. The other important thing Robert Hooke does is that as he sees these first cells, using what was originally supposed to be like a child's toy, a little telescope type thing, at this time, with again the Renaissance and art and science and all these advances, some of the first consistently well ground glass lenses were being used. And so he took this toy and began using it as a microscope, and many other people followed suit, and they started looking at all the samples they could find. And so this group said maybe his favorite color was brown because he's looking at cork. And somebody during first period came up with a very clever way to remember his name, that Hooke. After Hooke, other people were hooked on looking at cells because he started the trend. Okay? All right, so that's Robert Hooke. Then our next scientist is Anton von Leeuwenhoek. So let's jump ahead and learn a bit about Anton. So Anton von Leeuwenhoek, Dutch, with a big long name, he actually saw the very first living cells. And if you look at the date, so this is 1674, 10 to 15 years after Hooke. And so as everybody starts looking at things, he looks at lake water, pond water. So he saw the first single-celled living things, which he called animalcules, which I always liked that name just because it's very strange. It just means little teeny tiny animals. So he sees these little living things, just like you guys are seeing in your pond water, and he calls them animalcules. And then he looked at everything he could think of. He looked at samples of skin and hair and that scummy stuff off your teeth. And really, one of the other classes said maybe his favorite color was barf yellow because he looked at incredibly disgusting things, which he did. So the other way I remember, so these guys said you can remember because his name starts with an L, von Leeuwenhoek, that he saw the first living cells. The other way, and this is kind of a silly way, but the way I remember, Hooke and von Leeuwenhoek were the first two to use microscopes and discover cells. Hooke's name is shorter and simpler. He saw empty dead cells. Von Leeuwenhoek's name is longer and more complicated. He discovered living cells, more complicated cells, and he called them animalcules, which is also a kind of long, silly, complicated name. But that's the way I remember those guys. All right, so next we have Schleiden. So next we have Schleiden. He's German, and in 1838, after looking at many, many, many samples, and again, look how many years have passed here. So 74 to 1838, we've got what? Almost 50 years have passed, and many people looking at things. Based on his own research and other people's research, he says all plants are made of cells. So Schleiden, I think it's pretty tough to think of a way to remember that Schleiden discovered plant cells, but I think if we talk about the next guy, then I like grouping him in twos. Hooke, von Leeuwenhoek, Schleiden, and Schwann. So let's go to Schwann, and then we'll come back for a good way to remember Schleiden. All right, so now we have Schwann. And after much study, and again, and if you notice, the year is 1839, so it's almost the same time period. Well, it is the same time period, which makes sense. So these guys are all doing research and stuff at the same time. They come up with very similar hypotheses, pieces of the theory. So yeah, she said 1839. Anyways, but the way I remember Schleiden and Schwann, 1839. Again, if you chunk them together, Schleiden and Schwann discovered that all plants are made of cells, all animals. So Schleiden and Schwann, plants and animals, And Schwann sounds like swan. So that's a pretty easy way to remember that he's the animal guy, which means Schleiden has to be the other guy, right? But it helps me remember. So all animals are made of cells, and from there, we can kind of move on to all living things being made of cells, and we shall move on to our next scientist, who is Rudolf Virchow. So next group grows. Wrong one. Okay. So also around the same time here, Virchow comes up with the statement that that cells only come from other cells. And in fact, under the microscope, he actually saw cells dividing. So single cells reproduced by just basically making a clone. Saw cells dividing. Yeah, basically they copy the important stuff and then split into two. So he saw cells dividing and basically came up with even these simple single-celled living things come from other living things. They make copies of themselves in a way kind of similar or parallel to how animals reproduce. So Virchow, cells only come from other cells. So again, on the quiz, you'll have multiple choice. You got to match up the scientist with what they discovered. The fact of their different heritages, and we'll do the next two in just a second here, they all came from different places. We're now at, did you say 1855 for him? So we're now at 1855 to say that cells only come from other cells. So now we've gone almost 200 years from the first cells being found. So it's a lot of collecting of data. It's a lot of collecting of information. And many of these guys were not scientists as their first profession. Many of them did science just for fun as a hobby, but they had other careers that they did for a living. So let's move to our last two scientists, Francesco Redi and Louis Pasteur. Now, these two guys have a little bit different connection, and the textbook put them back in the previous section, which I don't like that they're in the other section, but we will jump backwards to look at them. We'll look at those tomorrow. So these guys, the big deal with these guys is that they helped to disprove spontaneous generation. They helped prove that cells come only from other cells. And I think the reason the textbook puts them in the earlier section is it also talks about the scientific method. But let's get our next scientist group to tell us a little bit about Francesco Redi and his experiment. So to fully understand Francesco Redi and Louis Pasteur, we first need to talk a little bit about spontaneous generation. Spontaneous generation was the incorrect assumption that flies and other living things would just come out of dead meat. Rags with rice would produce mice. That living things would just appear given the right conditions. And on one hand, this sounds absolutely ludicrous to us because, of course, it doesn't just magically come out of things. However, if you've ever in your kitchen had some fruit that gets a little too ripe, you know, an orange, a banana, all of a sudden these fruit flies just come out of nowhere, and they're so annoying. But things like that happen, and it makes me understand a little bit about how they thought spontaneous generation could have happened. It's the incorrect assumption based on observations. It's before people knew about microscopes, about microscopic cells. This was indeed in the 1600s and before then. And so people based things on what they saw and what they knew, on the stories people told. Okay? All right, so then Francesco Redi, as the group just explained to us, he disproved spontaneous generation. And the second thing he did that was so important is he didn't just say, hey, that's not right, because I know it's not. He actually had a controlled, repeatable experiment. And because he had a controlled, repeatable experiment, this really gets to be the very starting of the scientific method. And again, here we are in the Renaissance, all sorts of new breakthroughs in art and science and painting and perspective and all this. But controlled repeatable experiment. So he had these jars, and you'll read about this again and again in high school biology and college biology. He actually not just did this experiment once, but he tried it, and then he did a follow-up experiment, and then another one. And so he had the simple version that your textbook says. He has 2 jars of meat. One of them he leaves open to the air so the flies can land on it. And the second one, the top is covered with like something so the air can still get in and out. And the flies, this is a fly, the fly can land on this one and lay microscopic eggs. The fly can't lay on this one. So what he showed was if the fly can't lay the eggs, the meat won't grow maggots. And so yes, I remember him in a similar way. Ready, red meat, the rotting meat experiment. And yes, I like the Francesco, he is Italian. Francesco made a franchise to make refrigerators to refrigerate the meat so it wouldn't spoil, which is a made-up way to do it, so don't get confused. But, okay, so that's that. Make sense? Francesco, ready? And then finally, our last scientist does another similar experiment. Please tell us about Louis Pasteur. Let's go over that. So Louis Pasteur did a very similar experiment with broth. So this was in, you said mid-1800s? So in the mid-1800s, so you look at this, Reddy did this experiment at the very beginning of when cell theory was being discovered. Pasteur's follow-up experiments are in the 1800s. That's as cell theory is probably starting to become more popular. Now we have all plants, all animals made of cells. And so this is kind of an appropriate time because now we also know about the microscopic bacteria. So he did this experiment. And so broth, imagine chicken broth or vegetable broth, right? If you leave it out at room temperature, it's going to spoil, it's going to smell nasty, it's going to be disgusting. And Pasteur, his hypothesis was that there's bacteria in the air, on the surfaces of things, and that's why it gets disgusting. So he had, and the textbook shows you, but he actually had these special flasks, here's the broth, and the neck of it went like this. And it was just bent glass. And that way the air could get in and out, but the bugs could not. So basically he had 2 of these funny looking flasks, right, with broth in them. One of them he boiled, which sterilized it and killed all the bacteria. And the other one he just put regular broth in. And as he predicted, the unboiled one grew bacteria, got disgusting. And then he kind of followed up on this experiment by, after this one was, oh, yeah, look at that. After this one, after he'd proven his point, he snapped the lid off, snapped the neck off, so bacteria could get in from the air, and then it too grew stuff. So, and Pasteur, even today, his name is on something called pasteurized milk. Pasteurized milk, and actually almost all of the like juices, even all the fruit juices, they're pasteurized. And what happens is they go through a flash heat procedure, pasteurization, that kills a lot of that bacteria so that they have a much longer shelf life. So that then you can have, you know, your milk stay in the fridge. I mean, you still got to keep it in the fridge, right? But then you keep it in the fridge for a week instead of things spoiling very quickly. Yes? Okay, so then to finally kind of sum it all up and connect back to our first question, so now we have people from all over Europe. We've got people from Italy, France, England, Germany, and that the Dutch at this time were actually very, it was kind of a seat of scientific exploration up there in Holland. So we've got all of these different people over, starting in the mid-1600s and ending up with cell theory in 1855. So nearly 200 years of experimentation and sharing information, we get a theory. So that is cell theory. Take a look, test yourself over the different scientists, and see if you can remember who did what. look at your notes and other people's notes, and the quiz will be matching. Again, on the quiz, you don't have to know their first names, you don't have to know the exact dates, but just that big idea of the main thing each person contributed.